So this video is going to review Gametogenesis, the formation of gametes. And some of you might be thinking, well, didn't we already do that in class? We talked so much about meiosis. Meiosis 1, meiosis 2, homologous chromosome separation, sister chromatid separation. Didn't we do this already? And in a way, genetically, we did. The purpose of meiosis is to make gametes, to separate chromosomes in such a way so that cells can be created that contain half of a human genome. But there's more to it than just that. Male and female gametes are different. And in order to create the proper gamete in the right organisms, we need a more refined developmental pathway than just meiosis alone. We need to be able to create the right gametes in the right individuals so that fertilization can be possible. So consider this. Here is a female gamete, a female egg. And not surprisingly, it contains a genome, or a nucleus, with 23 chromosomes, half of a human genome. But that female egg also contains mitochondria for energy production. It contains a rough endoplasmic reticulum, as well as a smooth endoplasmic reticulum. It contains a Golgi body, and lysosomes, and peroxisomes. That female egg contains half a genome and all of the other organelles and cellular components that are needed to make a viable human cell. Now let's consider the male gamete. Like the female gamete, the male gamete contains 23 chromosomes. And that's it. Oh wait, it also has a tail. That's the male gamete. The male gamete is nothing more than a half-genome delivery device. It arrives at a female egg. The sperm head penetrates the egg, carrying its 23 chromosomes with it. Eventually, that migrates and finds its way to the nucleus, where there's fusion, and each individual chromosome are brought together as 23 homologous pairs, giving us our 46 total chromosomes and creating a new human life. So the male gamete needs to be constructed so it is a very durable and mobile half-genome delivery system. And the female gamete needs to be constructed in such a way that it contains as many cellular components as possible, as many mitochondria, Golgi, ERs as possible, because the larger and healthier that cell, the more chance the new life that will develop within it can survive. So with that context, let's think about gametogenesis a little bit more closely, but also remembering the purpose of it all, to make these two different types of cells. And let's talk about males first. We're dealing with humans, of course, so the very first thing is we start with a typical human cell with 46 chromosomes in it. Now, I shouldn't say typical, because this cell has differentiated to a point where it is destined to give rise to sperm cells. And for that reason, this specific cell is referred to as a primary spermatocyte. Now, I won't belabor the meiosis, because we've done that quite a bit. But this cell then enters meiosis, crossing over what happened in prophase 1. In metaphase 1, homologous chromosomes are lining up side by side, and they're being pulled apart to opposite sides of the cell in anaphase 1. And ultimately, at the end of meiosis 1, we have two cells with 23 chromosomes in them each. Now, when I say 23 chromosomes, do keep in mind that I mean chromosomes like this, chromosomes that are consisting of two sister chromatids held together at their centromere. And then we go into meiosis 2. In meiosis 2, sister chromatids are separated, pulled to opposite sides, giving rise to 4 cells that each contain 23 chromosomes. These 4 cells are referred to as spermatids, and the cells they derived from are called secondary spermatocytes. Now, although genetically these cells are what we want, they contain one of each chromosome, one member of each homologous pair, they're not yet viable sperm. They need to then develop a very tough exterior so that the DNA inside of them is protected. They need to also develop a very robust motor from which a tail can emerge and spin. And now these cells are competent for fertilization, and we can refer to them as sperm or sperm cells. And that's it for the male story. Your primary spermatocyte has a full human genome in it. Meiosis I is the reductive division. It gets that chromosome number down to 23 by separating homologous pairs and giving rise to 2 secondary spermatocytes. Meiosis II separates sister chromatids, giving us 4 spermatids, and then those spermatids further develop into sperm by gaining a hard exterior coat and a tail that allows them to swim and be motile. For females, the story is slightly more complicated. Just as in males, we start with an initial cell that has a full human genome, 46 chromosomes, and just as in males, we call this a primary, but in this case, we call it a primary oocyte because it's going to give rise to an egg. But we need to keep in the back of our mind that in this primary oocyte are all of these mitochondria that we want. Are the rough ER and the smooth ER that we need, the Golgi and the lysosomes and the peroxisomes and everything else that we need. So when this primary oocyte undergoes meiosis I, and those 23 chromosomes are given into 2 different cells, again, homologous chromosomes are separated in meiosis I, so our chromosomes look like that. We cannot have an equal distribution of cell material. We cannot have a typical cytokinesis, because if we did, we would have half the number of Golgi in each cell, half the number of mitochondria, half the ER, and we said from the beginning that we need the egg cell to be as large and robust and as full of cellular components as possible. So this just isn't going to work. Instead, at the end of meiosis I, we have what we refer to as an unequal cellular division. One member of each homologous pair goes into one cell, and the other member of each homologous pair goes into the other cell. But all of the mitochondria, all of the ER, all of the Golgi, and all of the organelles go into one single cell, a cell we call the secondary oocyte. This cell, which essentially is nothing more than 23 chromosomes, and that's it, a shell of a cell with no organelles in it, it is referred to as a polar body. In fact, it is the first polar body. And it's not viable. It's not competent for fertilization. It has no organelles. It's really just a holding place for one of each of the homologous chromosome pairs. And it dies. Therefore, the only cell that can undergo meiosis II is the secondary oocyte. And that secondary oocyte, once sister chromatids are pulled apart, gives rise to 2 cells, each with 23 chromosomes. And here, when I say 23 chromosomes, I mean chromosomes like this, what was formerly a sister chromatid. But this, too, is not going to work for the same reason as before. If we did an equal separation, a typical meiosis II cytokinesis, we would lose half of the mitochondria and half of the Golgi and half of the lysosomes in each of these cells. And we need our final cell to be as big and robust and as healthy as possible. So yet again, we have to pull off one of these unequal cellular divisions, where each of these 2 cells gets 23 chromosomes, But one of the cells gets all of the organelles, and the other cell remains devoid of them. You're probably not surprised to hear that this cell is referred to as our second polar body. It is not viable, it cannot be fertilized, and it dies. And this cell is referred to as our ovum. Just a few more changes and modifications have to happen to the ovum. Pretty much that's the development of a very specific sugary coat. This sugary coat protects the ovum, and much more importantly, it locks our speciation in place. Other mammals, other primates in fact, don't have on their sperm the right enzymes to chew through this sugary coat. The only species that has the specific enzyme needed to break down that sugary coat are human male sperm cells. So this keeps humans from mating with only humans, allowing no cross-species breeding to occur, which is good. It's good for our species' health. So once a sperm cell carrying its 23 chromosomes meets and fuses with a female cell, an ovum, carrying its 23 chromosomes, those 23 chromosomes can come together as 23 homologous pairs, or 46 total chromosomes, and make a new human being that is fully competent for survival because this cell has its ER, its Golgi, its mitochondria, all provided by the mother. So if we come back to that slide from the lecture, the one at the very end of the mitosis and meiosis lecture, we can see that this is exactly what we're trying to show here. These initial names, the cells that we initially start with, it's a little bit too complex, another level of complexity that we don't really need, so I cut those names out completely. We start the story with the primary spermatocyte. A single round of meiosis gets 2 copies of every chromosome down to 1. Essentially, we go from 46 to 23, and we have our secondary spermatocytes. Those secondary spermatocytes go through meiosis 2, where the chromosome number does not change. We stay at 23 and give rise to spermatids that can then mature into fertilization-competent sperm. For the females, we start with a primary oocyte as well with 46 chromosomes, 2 of each pair. But after the first round of meiosis, we cut the chromosome number in half to 23, but we need that secondary oocyte to be as large as possible, as full of organelles as possible, so we do an unequal cellular division and give rise to our first polar body, which dies. In the second round of meiosis, we keep the chromosome number the same, we're still at 23, but we also have to keep all of those cellular components in place as well, so we give rise to our second polar body, which also dies, and our ovum. And when a sperm cell enters an ovum, and the half genome of that sperm cell meets up with and pairs up with the half genome of the ovum, we have created a brand new and unique human being that's never existed on this planet before, our fertilized egg, our zygote, which will then, of course, undergo about 37 weeks of development and be born as a brand new baby. So that's Gametogenesis. If you have any questions, you know how to reach me, but hopefully it's clearer than it was before.